So remember, the 5' end comes first, and so you have to know the transfer RNA details, messenger RNA. Is it longer lived? Is it shorter lived? Does it get processed or not? Which one gets translated? Which ones are not? So please. The other yeah, you look at this. Excuse me. AUG is a start codon, true or false? I mean, the way I draw it here, AUG is a start codon, true or false? Why false? It's not true, actually. Why false?
Because long leading string is 3, right?
Yeah. No, I mean, if you go by this one, it's fine. Here. This is how it's going to be, because the taller one is the 3'. So this is 5' AUG. But it's not. Why? This is not a start. Remember, this is tRNA, guys. So AUG has got to be a start on the messenger RNA. So if you're trying to predict the messenger RNA, A will bond to U, U will bond to A, G will bond to C. What end will this be?
3'.
If this is the 5, this has got to be the 3. If this is 5, CAU. So remember, anticodon is tRNA, codon is mRNA. Which segues into the genetic code, which you have to know, characteristics. It's triplet. It's written in mRNA. It is non-overlapping. It is almost universal. It is degenerate. And remember, the definition of degenerate is what? One codon can represent many amino acids. That was a question, true or false? Because all of you took it for granted. No, it's false, right? Because one amino acid is giving you many codons, that's degenerate. One amino acid giving you many codons. If you have 5 bases and it's a quadruplet code, how many would you get? If it's 5 bases and it's a quadruplet code, how many codons would you expect? So here, these are the choices. I forgot what I said. You have 5 bases and it's a quadruplet code. So these are our choices: 5 to the 4, 4 to the 5, 4 times 5, 4 plus 5, whatever. Is it this or this? It's A, right? Because it's always, whenever you're in doubt, go back to the default. 64 codons. 64 is 4 cubed, not 3 to the 4. So if it's 4 cubed, this has to be the right choice. Know that there are exceptions, and they are mitochondria. Don't worry about the other organisms, mycoplasma. Or please remember, it's nearly universal. Let's see what else. Translation. You have to know the steps of translation, how many factors are involved, and also know the sequence, because you will be I forgot to mention it, but in DNA replication, same thing. Please remember the order of the proteins, right? Helicase, ligase, and so on. What's the first one? Helicase, other than the initiated protein. The last one to work is what? Ligase. True or false? DNA pool 3 works before DNA pool 1. Right? True. DNA pool 1 works at the end. Translation, you have to know, for example, if you have 1,000 amino acid long protein, what's the first one in bacteria always going to be? N-formal methionine, right? And true or false, the last one will be at the carboxyl end, right? Because you have N and C. Now, how many of those entered through the A site of the ribosome? right? All of them. So 999, whatever that number is. Okay. And then remember what's called collinearity. So, for example, what end of the mRNA corresponds to the carboxyl end of the protein? It's the 3, right? 3 rhymes with C, 5 and N. These are the leftovers. They don't rhyme. But, let's see. What's an open reading frame between a start and a stop? What? Then you have regulation of gene expression. Regulation of gene expression, know the difference between repressible and inducible, and then also be able to work out the details. So for example, the sugar arabinose. We did not discuss the ARA operon, but sugar arabinose, would you expect the operon to be inducible or repressible? Inducible. Why? If the sugar is not around, why make enzymes to break it up or break it down? So it's going to be an inducible. On the other hand, if you have the, let's say the vitamin 1, a vitamin, would you expect it to be inducible or repressible? In this case, if the vitamin is around, you shut the system down. Why? Because you already have it in place. And be able to know the significant hierarchy, the difference between the operator, the promoter, the structural genes. If one is mutated, right, what happens to the transcription? Is it on, is it off? So please be able to